Okay, let's cut right to the chase. Most students are literally throwing away easy marks on syllabus topic 1.2, and the crazy part is they don't even realize it until they get their grades back. You know the drill. You walk into the exam, you spot a question all about protein synthesis, and you write down what you think is this absolutely flawless, textbook-perfect answer. But there's a tiny, almost invisible trap hidden in the details of 80S and 70S ribosomes, and it completely tanks A grades every single year. So let's dive into this explainer and decode exactly what examiners are secretly hunting for so you can secure that top score.
So think about this seemingly basic question: where exactly are ribosomes located in the cell? I mean, it feels like an absolute gift of a question for day one biology, right? Well, that is the exact trap. Students read this, their eyes light up, and they write down something vague like, oh, they're floating in the cell, or they're in the mitochondria. And just like that, boom, the mark is gone. Examiners really don't care for vague descriptions. They demand precise, syllabus-specific terminology. So what should you actually be picturing in your head instead?
You've got to clearly separate the two distinct types of ribosomes. It's an absolute must. First up, we have the 80S ribosomes. These guys are the larger ones, and their exclusive home base is the cytoplasm of eukaryotic cells, where they're often attached to the rough endoplasmic reticulum. Then, in contrast, you have the slightly smaller 70S ribosomes. These are found exclusively inside organelles. We're talking specifically about the mitochondria and the chloroplasts. If you forget to attach that 80S or 70S prefix, or if you accidentally mix up their locations, it's an instant red flag to anyone marking your paper.
Now, you might be asking yourself, why doesn't just cramming the textbook over and over actually work? Well, ordinary revision totally fails for three very specific reasons: vague details, missing terminology, and completely ignoring the context. Cambridge examiners are testing whether you can transfer your conceptual understanding to entirely unfamiliar scenarios. If the exam hits you with a complex, totally weird setup involving a newly discovered eukaryotic cell, and you just regurgitate basic facts without applying them to that specific experiment, you are just throwing those points away.
But, okay, here's where we get highly strategic. In just a second, I'm going to reveal this hidden signal embedded in exam questions. It's a clue that basically whispers whether you're dealing with 80S or 70S without ever explicitly saying the words. But before we get to that, you really have to understand the foundational process that makes this clue so glaringly obvious in the first place.
Enter what we call the method mark strategy. When you're tackling a high-stakes, multi-mark question, like an experimental design or a heavy data analysis question involving ribosomes, you aren't just graded on stumbling across the final answer at the finish line. Not at all. You are evaluated on your step-by-step process. Practical work is all about specific skills: selecting the right apparatus, spotting anomalies, drawing conclusions based purely on evidence. If you structure your answer logically, then even if your final conclusion goes totally off the rails, the examiner can still throw you lifeline points for having a sound scientific method.
I really want you to listen to this warning, straight from the Cambridge Principal Examiner Report. Students lose marks by not showing working. Even if your final answer is wrong, method marks can save you. Let's say you get a question comparing the protein synthesis rates of 80S ribosomes in the cytoplasm against 70S ribosomes in a chloroplast under varying light conditions. Do not just jump straight to the end. If you just slap down the final number or a generic statement and it's wrong, you get absolutely zero, nothing. You have to prove your thinking, step by step.
So how do we lock down those method marks every single time? It's like a simple four-step cheat code. Step 1 is the concept. First things first, clearly state the core biological concept, like explicitly naming whether we're talking about 80S or 70S ribosomes. Step 2 is evidence or data. Literally pull the exact numbers, variables, or observations straight from the table or graph they gave you. Step 3 is scientific reasoning. This is where you connect your concept to the data. Ask yourself, why exactly is the 70S ribosome activity fluctuating? And finally, Step 4, examiner wording. Stick the landing by using the exact vocabulary from the syllabus. By moving through this sequence, you essentially force the examiner to hand over marks at every single level.
Okay, let's take things up a notch. What happens when examiners decide to throw statistical analysis into the mix with ribosome questions? It's a classic curveball, and you absolutely need a game plan for it. If you're handed a data set comparing the mean size of ribosomes extracted from two totally different tissues, you need to confidently state that a t-test is required because you're comparing means. Make sense, right? If you're dealing with categorical data, like, say, counting the presence versus the absence of 70S ribosomes across different genetic mutations, you've got to use chi-squared. And if you're tracking something like the rate of protein synthesis against shifting temperatures, well, that's a correlation test, looking for relationships between variables. Matching the right statistical test to your data, that is a top-tier skill for sure.
All right, I'm putting you in the hot seat for a second. Think about this statement. The mitochondria contains ribosomes for protein synthesis. I want you to mentally pause and really look at that. Based on all the traps we've just covered regarding Cambridge's super strict requirements, predict the flaw here. What is fundamentally wrong with this incredibly common answer?
Did you catch it? You absolutely cannot just casually throw the word ribosomes around. The corrected, examiner-approved top-band answer has to be the mitochondria contains 70S ribosomes for protein synthesis. That tiny prefix, 70S, is literally mandatory terminology. Without it, your answer is tossed out for being too vague. The examiner needs to see that you actually understand the massive evolutionary and structural differences between the machinery in the cytoplasm and the machinery hidden inside the organelles.
Let's do a really quick recap of the principal examiner's top 3 warnings so they're completely locked into your brain. Warning number 1, answers are too vague. Like we literally just saw, writing ribosomes instead of 70S ribosomes is a costly mistake. Warning 2, forgetting units and terminology. Always check your units if you're calculating sedimentation rates or physical sizes, and stick strictly to the syllabus terms. Warning 3, answers are not specific to the context given. If a question is specifically asking about a chloroplast, do not start rambling about animal cells in general. You have to adapt your knowledge to the precise scenario they put in front of you.
Look, when you're under that intense exam pressure, you literally need shortcuts. So here's a really quick memory hack to lock in these locations. I highly recommend using C80O70. It's super simple. Cytoplasm gets the 80S, organelles get the 70S. Just quickly jot down C80O70 at the very top margin of your paper the second the exam begins. It takes 2 seconds. But trust me, when you're 50 minutes in and your brain is starting to turn to mush, that tiny visual anchor will totally save you from a careless flip that could drop your entire grade.
But, here is the genuinely terrifying truth. Even if you permanently memorize C80O70, even if you flawlessly apply that 4-step method mark framework, and even if your biological knowledge of 80S and 70S is absolutely perfect, there is still one hidden cause that can completely derail your mark band. It all hinges on a single word right at the very beginning of the question. If you misunderstand the command word, literally everything we just talked about goes right out the window. Seriously, if you misread that command word, all your perfect 80s and 70s knowledge is useless. So ask yourself honestly, do you actually know the difference between an examiner asking you to evaluate a cell structure versus explaining it? Because if you treat them like they mean the exact same thing, the examiner just stops reading. To guarantee that A grade, you have to decode these exact instructions. So, what's your next move? Subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCSE to excel in your Cambridge examinations, and make sure you join us for the Command Words Masterclass, where we'll solve this final, absolutely critical piece of the puzzle.